Marco started his career, most of his career happened at the University of Torino in Italy, where he gets his bachelor in biotechnology, master in molecular biotechnology, and then his PhD in molecular medicine. And then he performed a postdoc with Judy Campisi, the Back Institute, which, well, she's a well-renowned scientist on the field of senescence. And, well, I guess Marco take the lead there. And then, well, he moved to Groningen University, where now he's an associate professor and chair in cellular aging. And he's, well, has done a lot of contribution in the field of senescence. He has a lot of papers in high impact journals, Nature, Molecular Cell Metabolism, EMBO, Aging Cell, and so on. So it's a real pleasure, and thank you, Marco, for being here.
Thank you, Cesar. Thanks for the introduction, for the invitation. Also, thanks, Christian, for inviting me. It's a pleasure for me to be here. Now it's morning for you, it's afternoon for me. So I will now share my screen. You need to enable me to share my screen because I'm not able at the moment. Yes, perfect. Now I should be. All right. I hope you see the full screen. Is it full screen?
Yes.
Yes.
Okay, perfect. All right. So yes. So what I would like to tell you today is indeed actually there are a couple of stories that are covering somehow this aspect that we are really interested to develop in the lab, which is going from understanding basic molecular mechanisms that are associated to senescence in order to develop also new interventions. And of course, this audience knows very well the concept of aging as essentially a condition that increases our susceptibility to develop diseases. Here you have a non-comprehensive list of chronic diseases that are indeed associated with aging, and going from neurodegenerative dysfunctions to cancer to cardiovascular diseases, etc. And also, we of course all know how these diseases, here you see some of the most common killers that we observe, heart attacks, cancer, stroke, and Alzheimer's disease, which are all exponentially increasing with age in humans. And this is true also for other organisms, but of course humans are the example where most of these chronic diseases develop and then eventually kill individuals. And also another important point before I jump into the real topic of the discussion is that of course we start from a point of view of interventions that is based on targeting specific diseases. So this is historically what we are trying to do in the clinic and in the preclinical environment to develop strategies to target specific diseases in order to delay that targeted diseases. But of course now we are also trying to shift a little bit the paradigm, especially in aging, by thinking that we can target basic mechanism of aging. And if we do so, and we are able to identify interventions that can do so, we can potentially delay multiple age-related diseases at the same time. Because of course, aging is indeed the condition with the highest association with a number of plethora of diseases. So targeting aging per se might indeed be a very effective strategy to achieve these multiple age-related disease intervention. And in the past decade or so, we had a better and better understanding of what are those basic mechanisms of aging that are happening at the molecular and cellular level. Here you see the famous wheel that was published in Cell almost a decade ago, where there are these 9 hallmarks of aging that are depicted. which all might function also as potential target for interventions that could cure or at least delay the onset of age-associated dysfunctions at the organismal level, of course. And what I want to discuss today is cellular senescence, which is indeed my personal interest and the interest of my laboratory. So in order to understand what senescence is, we need to start from how we develop and how we grow, in the sense that we all know that, of course, we start from being one cell. Of course, here can be a spermatozoa, but I got some critiques, and I should also use an egg to be politically correct. But anyhow, the starting point is one cell, where then this cell will grow. And during this growth, of course, this cell needs to replicate. And this to replicate a number of times, and then new cells will replicate also a number of times. Because we need to generate eventually trillions and trillions of cells in order to reach our full body maturity. But then, of course, this is not finished, because even when we reach this full body maturity, we still need to maintain the body size and the function of each organ and tissue in the body by regenerating the tissues, by turnovering cells all the time, which requires, again, the production of trillions and trillions of cells, which, of course, require a massive number of cellular replications that happen at each second in the body. And one problem with this massive replication is that somehow somatic cells have a sort of intrinsic clock that determine their replicative lifespan. So the cells are mortal in the sense that they can undergo only a certain number of cell divisions, and then at some point they reach a sort of crisis where they block their capacity to replicate. And this indeed is what we call the aphilic limit, which has then been defined as cellular senescence. And there are only a few exceptions to this aphilic limit. So cells that can escape this block of proliferation, and these cells are normally germline cells, very early embryonic cells, and of course, most of tumor cells, because most tumor cells are able indeed to grow unrestrained, and they don't have this limit. And we also know that one of the first indication and a molecular mechanism associated with this aphilic limit and this induction of cells in this permanent cell cycle arrest is the shortening of telomeres. Because we know that at each cell division, the cap of the chromosomes, indeed these telomeres, are shortening. And at some point, telomeres reach this critical length, which is recognized by the cell as a threat, and activates a cascade of DNA damage responses, which eventually make the cell to stop its ability to divide and makes the cell to activate these pathways that are associated with cellular senescence. And some of these blocks are actually the two most important blocks of two proteins, oncosupressor genes, which are p53 and p16, which are indeed very important mediator of the cell cycle arrest, the permanent cell cycle arrest, which is associated to senescent cells. And also it is important to note that after the description of senescence and state of permanent cell cycle arrest as a consequence of telomere shortening, additional stresses have been identified as a potential contributor to premature senescence, to the entrance of cells in this senescent state in an accelerated way. For example, we have genotoxic and oxidative agents. We have excessive mitogenic signals. We have several oncogenes and disruption to chromatin, which all can cause the aberrant activation of these responsive pathways that then eventually activate senescence mechanism. And also what we've learned is that senescence goes much beyond the simple cell cycle arrest. So senescent cells are characterized by additional morphological, functional, and structural changes. They are enlarged, so they have a different morphology than normal cells. They, as I mentioned, mainly are characterized by a DNA damage response and also changes in nuclear structure. For example, they lose the expression of this lamina protein, which is lamin B1. They activate or dysregulate a number of organelles. They have enhanced lysosomal activity. and content, which can be exploited by using the by measuring the activity of this lysosomal enzyme called also senescence-associated beta-galactosidase. They have dysfunctional mitochondria and signs of endoplasmic reticulum stress, which I know is a topic that is of interest of some of you in this audience. They have altered metabolism, changes in the plasma membrane. For example, they upregulate the expression of this plasma membrane protein called caveolin-1. And they also become resistant to apoptosis. So they upregulate mechanisms that are associated to anti-apoptosis. And this is a point which I will go back in a few minutes. And then there is another very important feature of senescence, which is the activation of the senescence-associated secretory phenotype, also known as the SASP, which includes a number of pro-inflammatory, growth stimulatory, and pro-angiogenic and migration factors. And all these factors can act either autocrinally by reinforcing the senescent state, or paracrinally by modulating the behavior of the neighboring cells. And now, of course, it becomes clear how senescence is a very complicated mechanism, because it can act by a cell autonomous mechanism, by restraining growth, cell growth, by blocking, of course, proliferation, but also in a non-cell autonomous fashion, because it can modulate the behavior of the neighboring cells via SASP secretion. And what we know also is that the SASP, having this very important and peculiar pro-inflammatory signature, can be an important cause or contributor to many age-related pathologies, which are indeed characterized by the presence of inflammatory markers. And it's quite interesting because age tissues are indeed characterized by what we call the sterile inflammation. Because they are inflamed, they have signs of inflammation, despite there is not a real pathogen that is causing this inflammatory response. And so part of the cause might actually be the presence of these senescent cells. And indeed, if we analyze the presence of senescence in young and aged tissues, we can clearly see that there is an accumulation of senescent cells with age. Here are some examples in human tissues. This is skin, colon, and brain, where if we monitor the presence of P16 positive cells, we can clearly see that there is a much higher presence of these cells in aged tissues, going from 55, 60 until 80, 85, 90, which are expressing indeed, which are more present, of course, in the aged tissues compared to the young ones in all these tissues. And this, of course, is also a non-comprehensive example, because there are many other tissues where senescence have been shown to be accumulated with age. And the same can be observed in mice. So we have now used here a transgenic mouse model that we have generated a few years back, actually a decade back. This mouse is called P16-3MR. It is a mouse where a 3MR reporter, which encodes for 3 proteins, a luminescent protein, a fluorescent protein, and a suicide gene, which can be activated when we give a prodrug called ganciclovir, under the regulation of the P16 promoter, being the P16 one of the main markers for senescence. With the idea that in this mouse, we can essentially monitor and usably eliminate senescent cells. And what Boshi did in the lab was, of course, to follow the presence of these cells in aging mice. Here you can see the example of the same animal monitored at different time points and the luminescence that comes from the animal. And you can clearly see that the luminescence increases with time. So the older the mouse gets and the higher the luminescence is. And also what we can see is that from an old mouse, now 24 months of age, we isolate, extract tissues, and we look at the luminescence. We can clearly see that there is a high luminescent signal from virtually every tissue of this aged animal, suggesting that indeed the accumulation of senescence essentially happens in every tissue. If I do the same job of monitoring luminescence in a young animals, I will not see almost anything, suggesting that indeed there is this accumulation of senescence in virtually, again, every tissue in an aged mouse. It is important to note that the percentage of senescent cells in the overall tissue is actually quite low, even in this condition, so even in aged tissues. So here you have 3 examples. You have the liver, the brain, and the skin, where you can clearly see that at maximum you reach around 4% of cells that are senescent, suggesting that indeed you don't need that many senescent cells, or actually you don't have those many senescent cells, but the number remained quite low. But of course, potentially these cells can be still detrimental because they secrete all these factors that can also affect neighboring cells. And indeed, what happens is that now if we do a therapy in these mice, where we eliminate senescent cells, so we activate our suicide gene now, providing the mice the prodrug, and we do this intermittently, starting at middle age. So every month we give a short cycle of elimination of senescent cells, and then after 6 months we evaluate the physical performance of these animals. Here I give you like very simple examples of physical performance. One is running, so how much the mouse can run, and the other one is strength, so the strength that the mouse have, measured as grip strength. And in both scenarios, in both cases, for both parameters, what you can observe is that if you now eliminate senescent cells in these animals, the animals maintain a better physical performance in terms of endurance and strength compared to mice that maintain senescence all this time, suggesting that indeed, even if you have this small percentage of senescence that accumulates with time, you still, these cells are still quite detrimental. And if you eliminate them, you can achieve a better health span. There is also an important concept to keep in mind that one thing is to accumulate senescent cells because of time, but this accumulation of senescence can be both accelerated and exaggerated by a number of different insults. For example, pollutants, radiation, drugs, diet, and injuries. So you can accelerate and exaggerate the accumulation of senescence if you expose them to these stresses. And here is one example using our 3MR mouse, whereas we expose young animals to genotoxic stress, either doxorubicin, which is a chemotherapeutic drug of the anthracycline family, or we give total body ionizing radiation at sublethal doses. And in both cases, we can see that even if these mice are young, because they're only 4 months of age, now you can see a very strong luminescent signal that is now developed because of after the exposure of to this genotoxic stress, suggesting that here you can accelerate essentially the burst of senescence by exposing the mice to this stress. And very importantly, what we observe is that these senescent cells are important contributor to inflammation. So now you have the case of doxorubicin, a young animal, and here you have the case of the plasma and the kidney, whereas the exposure to this doxo, to the genotoxic stress, increase all these pro-inflammatory markers, such as pro-inflammatory interleukins, chemokines, and others. And now, if you remove the senescent cells that are generated by the exposure to the genotoxic therapy, you reduce the inflammation in these mice, suggesting that indeed the senescent cells are not only generated, but they're also producing all these pro-inflammatory markers that somehow can be detrimental in the mouse because they can cause a state of chronic inflammation. And indeed, also in this context of accelerated, essentially, senescence, we can observe that if now we remove these senescent cells that are generated by the chemotherapy by exposing the mice to the prodrug that work in our transgenic animal, and we again monitor the same physical activity of the mice by the running that the mice have and the strength they have, we again observe that while the exposure to the chemotherapeutic treatment really affect their health, and the mice now run less and they're weaker, if we now eliminate the senescent cells that are generated by the therapy, the mice are performing much better. So suggesting that indeed, also in this context, the presence of senescence is detrimental. And then we also did look into age-associated dysfunction, in this case disease, which is cancer. In this case, what we've done is that we have implanted breast cancer cells into the mammary fat pad of these P16 TMR animals, and then we have exposed them to doxorubicin or to a combination of doxorubicin and acyclovir to eliminate all the senescent cells that are generated in the body of the animal besides the tumor. Because of course the tumor does not express our transgene, so even if senescence is induced in the cancer cells, they will not be eliminated. So here is just to study the effect of systemic senescence that is present in the animal on an age-related disease, which is cancer. And indeed, then we follow survival and metastasis in these animals. Now I'll show you the image of the metastasis because these cancer cells are labeled with a luminescent reporter, which is different from the one that we have in the transgene. So then we can monitor specifically also the spread of these cancer cells. And what you can see is that while in an animal that is exposed to doxo only, there is a period of dormancy where the tumor, the primary tumor stops. But then this tumor relapses after some time, and it goes to colonize the liver and the lungs, forming metastasis. And this happens in around 80% of the mice. Now, in the group that has been eliminated of senescent cells because of this prodrug, the number of animals that develop metastasis is much reduced. And for these, the mice actually live longer than the mice that see the chemotherapy only. Suggesting that indeed, if you have senescence in the environment, now these senescent cells are able to promote tumor recurrence and affect, of course, survival of the mice. Then another example of what can happen on an age-related dysfunction using a model for the disease is Parkinson's disease. So this is an example where we have used a pollutant, which is called paraquat. It's an herbicide, which is still used, especially in California. And it's been observed that this pollutant, this herbicide, is quite toxic, especially for neurons, dopaminergic neurons, and it's causing this Parkinson's disease-like pathologies, motor dysfunction and depressed neurogenesis. And is used now also in preclinical models in mice to indeed study Parkinson's disease. So what we observe is that if we expose mice to this herbicide, now the mice develop senescence in the brain, and in particular, they develop senescence in astrocytes. So senescent cells, senescent astrocytes arise as a consequence of this exposure. But what is very interesting to see is that now if we eliminate these senescent cells that are caused by the exposure to paraquat, we improve neurogenesis in the subventricular zone in these mice, and as a consequence, the phenotype is ameliorated. So now you can see an example of the rearing, so how a mouse would now try to escape this beaker where it's put. Here you see the mouse that has been exposed to paraquat, which is just sitting at the bottom of the beaker, and it doesn't really try to find a route to go out. But now if we eliminate senescent cells, now this mouse is much more active and is trying to get out from the beaker. Suggested that indeed the presence of senescence induced by this oxidative stress, which is paraquat, can really sustain the development and the progression of this Parkinson's disease-like pathology. Then a third example that I wanted to show you is one of the most detrimental age-associated dysfunctions that we have in humans, which is osteoarthritis. And in this case, what we have used is a model where we have post-traumatic osteoarthritis, whereas this is a surgical procedure where we essentially induce these osteoarthritic-like joints. In particular, we do this on the knee of the animals, of the mice, where essentially this osteoarthritis is associated with excessive loss of cartilage, where then the bones are actually essentially frictioning against each other, causing the mouse to have pain and not to use that operated leg anymore, or at least much less than the non-operated one. So what we see is that when we do this surgery, senescence slowly appear. Here you see the luminescence coming out from our reported mouse. That is in the knee. It is much higher luminescence in the knee that has been operated compared to the sham knee. So the knee that has been just open but not went under the surgical procedure. And that we can also eliminate actually these senescent cells if we expose now our animals to the prodrug. But what is very interesting to observe is that if we do so, we actually ameliorate again the degeneration of the knee. So here you can clearly see that when we expose the mouse to the surgery, we lose cartilage, which is this orange-red layer here. This is a normal condition, and this is the mouse that has been operated, where you see that this layer of cartilage is extremely thin now. And now we see that the bone essentially the two bones touch each other, causing, of course, pain. But here, if we the condition where we have eliminated senescent cells, where we can clearly observe that there is a much better layer of cartilage that is preserved, and that, as a consequence, the mouse does not feel that much pain and is using that leg as much as the non-operated leg, suggesting that indeed the mouse does not want to, does not feel that pain that it would observe or feel if it was only operated without that going with the senolytic therapy, so the elimination of senescent cells. And here you can see that also the same we can observe some amelioration of the phenotype when we essentially ask the mouse to retract from a hot plate. And again, the operated leg is slower to be retracted because the mouse is really not able to use the leg much. It takes longer to retract from the pain of the hot plate, but the mouse that have been eliminated of senescent cells now can respond much better. So there are, of course, many other now indications where senescent cells have been associated and where they have been actually shown to be either contributing either to the onset or to the progression of these diseases. I'll show you some examples about neurodegeneration and cancer, but there have been examples in metabolic disorders, in fibrotic diseases, and you name it. This list is growing overnight, and it's difficult to keep track of how many diseases have been associated to the presence of senescent cells. And for this reason, of course, one of the things that we essentially would like to do is that we would like to intervene against these detrimental senescent cells with a therapy that can potentially be used in humans, because all the examples I have shown you so far have been derived from a transgenic animal. And despite, my hope is that we will have transgenic humans at some point, it is still not a feasible approach. So we have to find alternatives to this. And alternative to this is indeed the study of potential xenotherapeutics, so drugs that could interfere with detrimental senescence in order to preserve health and potentially improve longevity in humans. And there are a number of different options that you can use to achieve these xenotherapies. But there are two particular classes of xenotherapeutics that I would like to describe a little bit more in depth. And these are Synalytics, which are essentially drugs that can selectively induce senescent cells to die, and xenomorphics, so drugs that can modulate some senescence-associated phenotypes. And in particular, among those, the most interesting one seems to be drugs that can inhibit the SASP, because the SASP being potentially responsible for most of the detrimental function of senescent cells. So the first example goes on Synalytics, and it actually starts from the idea that senescent cells are somehow heterogeneous. And there are different intrinsic and extrinsic factors that can affect how the senescence phenotypes develops and mature. And these indeed are depicted here. This is, again, a non-comprehensive list of all the potential factors that can determine and influence senescence cell phenotype. But we did in the past a study, and this was pioneered by a PhD student in lab, Alejandra Hernandez Segura, which decided to consider many different variables, many different of these factors, put them all together and try to see if there is a core signature in the senescence phenotype. And in particular, she focused on the transcriptome of senescence, so of the different subset of senescent cells, to understand whether there is this common transcriptomic signature. And eventually, she derived this list of around 55 genes, which are somehow equally distributed among downregulated and upregulated genes. Whereas we don't find the usual suspects that have been associated previously to senescence, but where we can clearly identify genes that totally make sense in the senescence phenotype. Because if we then look at categories that describe these genes, we can see association to cell cycle regulation, which is, of course, one feature of senescence. We can see genes that are related to p53 regulation, which, of course, is another important feature in senescence. inflammatory responses, and of course, apoptotic modulation. I mentioned earlier that one of the features that we could observe in senescent cells is indeed this activation of anti-apoptotic mechanism. And indeed, several of the genes here somehow are regulating anti-apoptosis or at least protecting cells from apoptosis. It is quite interesting because it seems that senescent cells essentially are primed to apoptose, but then they also mount resistance to this priming by overexpressing anti-apoptotic genes. And some of these genes are members of the BCL2 family. For example, you can see one here, which is BCL2L2, also known as BCLW, which is highly conserved in indeed different type of cells. So we reason, of course, that perhaps if we try to interfere with this anti-apoptotic mechanism, for example, by using a drug which is called ABT263 or navitoclax, which is a pan-BCL2 inhibitor, we can potentially achieve senolysis, so the selective elimination of senescent cells. And indeed, what you can see here is that when you expose normal cells, non-senescent cells to ABT263, nothing actually happens. But now if we do the same with cells that are senescent, either because of ionizing radiation or because of replication stress, now you can completely almost completely eliminate these cells by exposing them indeed to this pan-BCL2 inhibitor. And important to note that this was done in collaboration with Dao Zhou, which at the time was at the University of Arkansas and now is at the University of Florida. And similarly, you can achieve the same if you go in vivo, you go in mice. Here is our 3MR mouse, and this is our doxorubicin-induced senescence model, where you can clearly see that you have luminescence when you expose the mice to doxo. You can eliminate senescence if you treat the mice with our prodrug, which is acyclovir. And now you can achieve the same downregulation of senescence if you expose the mice to ABT263. So also in mice, this works. However, as I mentioned, not always and not all the time, senescence is a homogeneous phenotype. So we also reason that perhaps there might be conditions where this strategy doesn't work, or that maybe the anti-apoptosis work via other mechanisms. And one of these is actually an example that I wanted to show you is the example of senescent melanocytes. So initially we were interested in studying senescent melanocytes because they populate nevi of moles. You can clearly see that these moles are essentially nests of senescent melanocytes. So we reason, and actually this is work pioneered by a postdoc in the lab, Jaskar and colleague, that perhaps we can use senolytic strategies to reduce nevus size. So we can maybe just apply a cream that contains a senolytic agent on the nevus, and then we can maybe make the nevus shrink. So we started by using ABT263 in just ex vivo senescent melanocytes. And what we could observe, first of all, was that, well, these senescent melanocytes express, upregulate these BCL2 anti-apoptotic proteins, in particular this BCLW. However, they are almost completely resistant to the treatment with ABT263. So there is just a small percentage of senescent melanocytes that are sensitive to the treatment with ABT263 or to the treatment with another analog of ABT263 called 736. So then we went back to, let's say, basic biochemistry on how apoptosis is induced. And we realized that, of course, on one side, apoptosis can be prevented by this anti-apoptotic family of BCL2 proteins. But on the other side, apoptosis can be also blocked if there is an overexpression of this other anti-apoptotic protein, which is called MCL1, which is an inhibitor of a pro-apoptotic, a very pro-apoptotic factor, which causes cytochrome T release, which is called NOX. And what Jaskaren found was that in senescent melanocytes, the exposure to ABT263 was sufficient to now have upregulation of this MCL1 protein. So essentially, while we were trying to block the BCL2 family, on the other side, the cell was trying to resist apoptosis by upregulating this MCL1 protein. And then we reasoned that if we blocked MCL1, we might now achieve senolysis. And indeed, if we now take these senescent melanocytes, we expose them to ABT263 and to a small short terpene against MCL1, now we achieve a very strong senolysis. So now the cells are almost completely dead. So of course, this was interesting, but we also wanted to see whether we could achieve the same with a drug. And luckily enough, a couple of years ago, a new MCL1 inhibitor, a very potent MCL1 inhibitor, was developed. It's called S63846. So then we combine ABT263 with this MCL1 inhibitor first in different type of senescent melanocytes. And we could see that in all cases, there was a strong senolysis in the cells. So a strong toxicity of the combination between ABT263 and the MCL1 inhibitor in the senescent melanocytes. However, these drugs indeed alone were not really effective in killing them. So there is a very strong synergy between these two compounds. So then, of course, our original point was to try to see whether this strategy of eliminating senescent melanocytes, a potential strategy to eliminate senescent melanocytes, could work in an in vivo setting of nevi. And to do so, we use a mouse. This is a mouse, it's called BRAF V600E. This is a mouse where there is this oncogenic mutation in BRAF, which can be inducibly activated specifically in melanocytes because it's under the regulation of melanocyte-specific promoter. And when you activate this oncogene, essentially you cause some sort of nevi in the mouse because there is an hyperproliferation of melanocytes. And then as a consequence of this excessive mitogenic signal, the cells stop and block in senescence. But before doing that, they indeed form these colonies, which are now pigmented lesions that can be recognized in the skin of these mice that you can see here because they are stained for melanin, which is, of course, this black pigment that is produced by these senescent melanocytes. And very interestingly, what happens is that now we expose these mice with this nevi, with this hyperpigmented lesion, to our combinatorial treatment. We can achieve almost complete disappearance of these lesions because indeed we are eliminating the cells almost specifically. So there is a reduction in nevocytes and hyperpigmentation just because we are exposing these mice to this combinatorial treatment and because we are now, of course, eliminating the source of this pigmentation, which are senescent melanocytes. So, of course, these are only a few examples of Synalytics, and if you follow the field, there are now a number of other Synalytic approaches that have been developed. You can see here, again, a non-comprehensive list of all these Synalytic approaches. But essentially, 99% of these approaches are going towards trying to reinstate apoptosis in senescent cells by twisting somehow this resistance to apoptosis that senescent cells can mount in different ways. So the strategies are always quite similar. And then at last, I wanted to give you an example of another strategy, which is based on inhibiting the SASP and not now to kill senescent cells. And this inhibition of the SASP started from a project that we were developing in order to understand how senescence behaves in different organs that are exposed to different oxygen conditions. So you might know that the partial oxygen tension is very tissue dependent. So different tissues are exposed to different type of oxygen concentrations. For example, in the liver, we have an average oxygen of 5%, which is what we call a normoxic condition because it's actually the average that we have in the body. But we have other tissues, such as certain part of the skin, where now we reach a condition of a concentration of oxygen, which is much lower, it's around 1%. And these are what we call physiologically hypoxic regions. Interestingly enough, we also have tissues, such as the kidney, where we have an intra-tissue gradient of oxygen, where we go from higher to lower oxygen concentration, from more normoxic up to actually 9% of oxygen to more physiologically hypoxic, down to 1% oxygen in the same one. In the cortex, you can see exposed to high oxygen, and the medulla, which is exposed to low oxygen. So what Tijmen van Vliet, a PhD student in the lab, did was to initially take primary fibroblasts, inducing them to senescence, and then see how these cells were behaving in either physiologically hypoxic condition, 1% oxygen, or normoxic condition, 5% oxygen. So what Timon could observe was that the capacity of these cells to enter irreversible growth arrest or to activate markers of senescence such as the lysosomal enzyme beta-galactosidase or the DNA damage, chronic DNA damage response, was similar in the two oxygen concentrations. So there were actually no differences in how the cells enter this senescence state. However, what was really striking was that what Timon observed was that now the cells that were exposed to 1% oxygen develop a much milder SASP, so a much lower production and secretion of pro-inflammatory SASP factors, pro-inflammatory interleukins and chemokines, which could be observed both at the mRNA level and at the protein level, suggesting that indeed hypoxia can restrain the SASP, at least in culture. So then we went back in vivo, and we looked at first at aged mice, 24 months of age. And here I give you the example of what we observed in the kidney, which is indeed this tissue where there is this natural gradient of oxygen, where it was very interesting to see that while the number of senescent cells that were developing in the cortical or the medulla area in an aged mouse were similar, so there was an upregulation in both areas compared to a young animal, which was indistinguishable. So both areas developed the same number of senescent cells. Now, the presence of SASP factors, which was much higher in the normoxic area, so the area that is exposed to normal oxygen concentration, compared to the area that is exposed to physiologically hypoxic level. So there was more SASP in the cortical area compared to the medulla area. And to note, we did these stainings in collaboration with Vasilis Gorgoulis at the University of Athens. Then we also did the same, but now looking at humans. So we took now, thanks to a collaboration with the nephrology department of my institute, we took old kidneys from old individuals, post-mortem, and we analyzed the same. So the expression of senescence, and here senescence is, again, sorry, I didn't mention before, but is actually measured as the upregulation of lipofuscin, which is one of the lysosomal markers that is associated with senescence. Or we stained for SASP markers, interleukin 6 and CXL1. And what we could observe again was that there was an equal number of senescent cells in the cortical and medulla area, but again, the SASP was much more pronounced in the area that is exposed to higher oxygen concentration compared to the area that is exposed to lower oxygen concentration. So then we went on and we tried to understand what was the potential mechanism by which hypoxia could restrain the SASP. So to make a very long story very short, we identify the AMPK-mTOR axis as essential for this control of the SASP. Whereas during hypoxia, there is an activation of the AMP kinase. And the AMP kinase is an important repressor of mTOR activity. And mTOR is essential to establish a feedback loop with NF-kappa B to promote the production and secretion of various SASP inflammatory factors. So blocking mTOR is also then blocking or at least restraining the production of the SASP, at least of the pro-inflammatory part of the SASP. So then what Timon asked was, well, can we achieve a similar result, so a SASP repression, by maybe using drugs and using drugs that can mimic hypoxic condition? So then we, of course, we selected a couple of hypoxia mimetic compounds which are available. And here I show you one of them, which is quite famous. It's a quite famous compound. It's called roxadustat. It's an approved drug for the treatment of kidney anemia, but it's more famous because it's been used as a doping drug, because it can enhance the level of erythropoietin. It has been used by cyclists to indeed boost their erythropoietin levels. So what Timon did initially was to take again our cell culture model of senescence and expose now cells that are at 5% oxygen, which are having this robust SASP, to the hypoxia mimetic compound roxadustat. And what he could observe was that the exposure to roxadustat was sufficient to decrease an already established SASP, suggesting that indeed we could block this exacerbation of the SASP simply by indeed achieving this block of the mTOR pathway, which indeed we proved the mechanism by which this drug works. So then we went back in vivo and we tried to treat our 24-month-old mouse with roxadustat. Again, we went with a few intermittent treatments with this drug. And we monitor the SASP, we monitor mTOR activity, and we monitor physical performance. Here again, I show you the example of strength. And what we could see was that in tissues, the liver and the cortical area of the kidney, there was, after the exposure to this hypoxamimetic compound, there was a decrease in the SASP. This was associated with a decrease in mTOR activity, and this was now associated with a better performance of these mice in terms of strength. So now we could also achieve a better health and health span simply by exposing the mice to this compound that was able to reduce the SASP in tissues. And again, now there have been developments of different types of SASP inhibitors. Again, a non-comprehensive list of them is depicted here. But most of them work either by modulating the mTOR or the NF-κB arm, which are indeed very important to activate SASP production and secretion. So with this, I will conclude by this summary slides. I hope that I could show you that senescent cells accumulate during aging and stress, and they are a contributor to pathology and tissue dysfunction. I also show you that the senescence program is heterogeneous and can depend on various intrinsic and extrinsic factors, but also that the understanding of the senescence heterogeneity can be exploited to potentially generate pharmacological interventions that counteract their detrimental function. And of course, the most important slide, which is the one of acknowledgement. I think I mentioned most people that contributed to the projects that I show you, and of course, a big thank to the funding sources and to you for your attention.